Hello, my name is Daniel Farb and I will be presenting this poster on an experiment entitled Expression of polyQ in Drosophila melanogaster causes a decrease in motor function. Huntington's disease is an illness that can cause different kinds of symptoms such as decreased motor function and cognitive function, among many others. The disease is caused by a higher than normal number of CAG repeats in the Huntingtin protein. Q128 means that the Huntingtin protein contains 128 CAG repeats. The experiment used two different genotypes of flies, male Q128 flies and female Q128 flies. The females have an LV driver, which allows Q128 to be expressed, whereas the males do not have a driver and therefore do not express Q128. The males are used as the control group in this experiment because the only main difference between these two genotypes is the expression of Q128, which is what we are studying. Figure 1 shows what Drosophila melanogaster, the model organism for this experiment, looks like. This experiment was performed using a negative geotaxis assay to measure the motor function of each fly genotype. This was done by placing the female flies into the graduated cylinder, tapping them down until they got to the bottom, then releasing them and counting how many flies reached the 35 centimeter mark in 45 seconds or less. This was repeated for the male flies as well. Figure 2a shows what it looks like when the flies are tapped down to the bottom of the graduated cylinder. Figure 2b shows what it looks like when the flies are traveling up the graduated cylinder. Since the two genotypes had a different number of flies, the percentage of flies that reached this 35 centimeter point in under 45 seconds was determined. These percentages were averaged over the 6 trials. Figure 3 shows that on average, 77.8% with a standard deviation of 9.0% of male flies reached 35 centimeters in 45 seconds, while only 9.1% with a standard deviation of 5.9% of female flies crossed the line. These results show that a much higher percentage of males reached the indicated line compared to the females. This result makes sense since we were expecting the female flies to have decreased motor function due to the expression of Q128, causing Huntington's disease. The next step in this study is to improve this experiment by doing two things. One, making the tapping down motion uniform by using an apparatus that keeps consistent the number of times a vial is tapped down as well as the force with which it is tapped down. And two, additional trials will be performed to increase the sample size. I'd like to acknowledge Paul Berkson, my lab partner, the TAs, Dr. Lindsay Mermanesh, Dr. Keen Piasta, as well as Professor Melissa Kozinski-Collins for accompanying us along every step of the way in this course. Thank you.